time for This Week in Virology, episode number 234, recorded on May 19th, 2013. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Today, we're coming to you live from Denver, Colorado. We're at the annual meeting of the American Society for Microbiology. And with me today is a TWiV co-host from the University of Michigan, Kathy Spindler.
Hi, Vincent.
Welcome.
Thanks.
As Elio said, we're in vivo.
We are in vivo, and these are really nice digs, a lot nicer than my office chair.
What brings you to ASM?
Well, I'm the division chair-elect of Division S, which is the DNA viruses division.
What's a division?
A subgroup of microbiologists in the ASM. I'll learn more about it after our post-meeting meeting on Wednesday.
Sounds complicated.
Yes.
So what we have done is to pick two speakers from this meeting. There are lots and lots of them here to pick from. As Elio says, it's like being in a candy store, not being able to decide what to pick. But we have two virologists here to join us and talk about their work. First is a professor in the Department of Molecular Biology at Princeton University, Tom Shenk.
Hello, Vince.
Welcome to TWiV, and I understand it's your first podcast ever.
This is my very first podcast, and I think I'm glad to be here.
It's like having a first child. Never leaves you. Also joining us today, he's a professor in the Department of Human Genetics at the University of Utah, Nels, Elde. Welcome.
Thanks, Vincent. Good afternoon.
And I watched your video today, so I knew how to pronounce your name.
Appreciate it very much.
Welcome to TWiV.
Good to be here.
And both of you are speakers, and you've both spoken today, correct?
This morning.
All right.
Yep.
Now, before we get into what these two do, I want to tell everyone that there are some ways that you can use to send us questions. If you are on Twitter, you can use the hashtag ASM2013, and that will bring it to our attention, and we can answer your questions here. And of course, the thousands of you in the audience can go up to the mic. You'll have to line up, and we don't know if we can get to your question, but there's a mic here, and you can ask your questions. We have an audience here. This is great. Do you guys know there's a virology session going on somewhere in this building? Yeah, but this is cooler, right? Don't tell them I said that. All right, you got the weather?
We have the weather.
What is the weather, by the way?
The weather is 66 degrees, and they show a little lightning for this afternoon, but I don't think it's started yet. But there has been rain in the forecast for today. It's very nice in Denver. We're right downtown. There was a marathon this morning. It looked like it was really beautiful weather for that.
Really? Did you go out and watch it?
No, but I knew it was in the way of maybe us getting here from Boulder. So.
All right, great.
I just checked the weather in Salt Lake just to get into the spirit of TWiV here, and it was 63 degrees Fahrenheit. That's about 18 Celsius.
All right. And did you check the
I didn't
weather?
check the weather in
All
New
right.
Jersey, sorry.
Next time you're on TWiV, you can do that. All right, let's start with you, Tom, since this is your first podcast. And I've known you for many years. And
Yes.
Early in my career, well, let's start with this first. Where are you from?
I was born in Brooklyn, but I grew up in northern New Jersey.
Brooklyn. Another scientist from Brooklyn. There's so many.
But I left there when I was 5.
You grew up in North Jersey?
And I grew up in North Jersey because my father worked in Manhattan. And one day he came home and said, I got this terrific promotion, and so we're going to move to New Jersey. And I don't know why we did, but we did.
What town was that?
So I grew up in a town called Bergenfield,
Okay.
not far from the George Washington Bridge, which of course ain't far from your digs at Columbia.
Yeah, I know Bergenfield quite well. So you went to high school in Bergenfield, too?
I did.
And college?
I went to a small college in Detroit called the University of Detroit.
Okay.
And I went there because it's an engineering school, and I was going to be an engineer. And after one semester of engineering, I realized I was going to be a biologist.
So you didn't like engineering?
I didn't do well with engineering.
And then right out of college, did you go to graduate school?
I did. So I moved back to New Jersey and went to graduate school at Rutgers. In fact, at the time, I joined a brand new department of microbiology at Rutgers Medical School. And the department chair was a fellow named Walter Schlesinger. And I worked for a fellow named Vic Stoller.
Right.
And Vic has been a mentor since that very first day of my experience as a graduate student. And he's just a terrific fellow. I've learned a lot from him.
So he's a virologist, so that must have been
Of course he's a virologist.
your entree into virology, right?
That's right.
What virus did you work on?
As a graduate student, I worked with Synbus,
Okay.
which all of us virologists know is an RNA virus. And
The best.
Of course, I discovered at the time, this was before you guys had cloned everything, that it's not easy to work with RNA viruses.
Right.
And so I then, as a postdoc, went out to Stanford, worked with Paul Berg, and switched to DNA viruses, which I
Okay.
found a lot easier to work with.
Now, when you were in the Berg lab, did you know Steve Goff?
I sure did. Steve Goff was a graduate student at the time.
Okay, 'cause
And he was a real mover and shaker in the lab.
he is my neighbor at Columbia now. He's my office mate right next door to me. So,
Well, I think very highly of Steve.
all right, so that was your postdoc. And then, if I remember, your first job was at UConn, is that right?
That's right.
Pretty good, huh?
At the UConn Medical Center
Right.
in Farmington, Connecticut.
So when I was a student, I remember reading your papers out of UConn.
You do?
I do.
Wow.
And you worked on adenoviruses?
Yes, I went to UConn and I began to work on adenoviruses. You know, we had, at the time, these new things called restriction enzymes. And so I had, with Paul Berg, been using restriction enzymes to do some of the very early manipulation of the SV40 genome. And then when I moved and I found myself on my own, I decided, hey, I'm going to try a different virus. And so I started to work with adenovirus. And we managed to make some mutants using restriction enzymes.
Mm-hmm. And then, how many years did you spend in Connecticut?
I was in Connecticut for 5 years.
Okay. And you moved to Princeton?
No,
Stony Brook, right?
I moved to Stony Brook.
Okay.
Arnie Levine had gone to Stony Brook as the chair of the microbiology department. And I had come to know Arnie fairly well from meeting him at adenovirus meetings.
Mm-hmm.
And when he landed in Stony Brook, he invited me to come and give a seminar. And I found that this was a very exciting, newly building department at Stony Brook, and I wanted to be part of it. So
Right.
I moved and joined Arnie and people like Eckhard Wimmer.
Right.
In
My
the
good friend Eckhart, yeah. And you spent many years there, and I know now you're in Princeton, so at some point you decided to move there. And that's your career, right?
Up to now.
I think we should ask Els about his career. Where are you from? You're from Minneapolis, is that right?
Yeah, I am, and I saw you were just there
I was there
not
last
too long
week.
ago, yeah.
Yeah,
So I
coincidence, right?
guess so, by definition.
But you didn't go to the University of Minnesota.
I didn't. I went to a smaller liberal arts school about 45 minutes south, Carleton College.
Okay. And were you a biology or an engineering major? Or?
I was deciding between biology or psychology, and it swung towards biology when I noticed in my psychology classes I kept trying to bring biology into my projects, and so that kind of tipped me off that maybe that was the right direction to go. Yeah.
But in biology, in research, it's good to have psychology background, right?
Well, yeah, and it's interesting. So my mom is a psychologist, family therapist. My father is a scientist, but I think just starting my lab recently, the psychology background, it might be more important on a day-to-day basis in some
right.
ways than the biology
That's
background.
right. That's right.
Yeah.
And where did you do your PhD?
So I took a little bit of a detour after graduating from undergrad. I was a scientist at a medical device company for a few years called Medtronic in Minneapolis. And then from there, I applied and ended up going to grad school at the University of Chicago. So, and I was in the Department of Molecular Genetics and Cell Biology there.
So did you take a little hiatus because you weren't sure what you wanted to do?
You know, I would say, a little bit burned out from the undergrad years and just kind of wanting to catch my breath. I actually worked for 1 year as a technician and then 2 years as a scientist. And it was a great way to just kind of step back from everything and kind of take
Right.
stock of things, and then I kind of realized that, no, getting back into academics felt like the right thing to do.
Right.
And so I did.
Now, if I remember at Chicago, you worked on tetrahymena, is that
I
right?
did, yeah, the model ciliate tetrahymena. So at this stage, I was really interested in model systems genetics. And this kind of had come from my time at Carleton even. And so all my rotations were in model organism labs, so a Arabidopsis lab, a yeast lab, and a tetrahymena lab. And that's the one that in the end kind of really caught my fancy and I ended up joining. So
Okay.
yeah.
Now, we've talked about Nels's work on TWiV before. and-
TWiV 198, I believe.
Oh,
Wow. What was 132, do you remember?
I don't, but I went back and looked at 198, so I'm pretty sure that's it.
And so we know that you were a postdoc with Harmit Malik.
yes.
And how did you decide to go there? What was the impetus?
Yeah, so I was doing kind of straight model systems genetics and cell biology.
Yeah.
But in, as you pointed out, tetrahymena, the model ciliate. And so if we think about the evolutionary divergence between, say humans and yeast and humans and tetrahymena, it's not even close. So our last common ancestor was hundreds of millions of years earlier. And you can't kind of help but start thinking about, well, what's the evolutionary sort of implications of this? And I was at Chicago, which has a great tradition in evolutionary biology. And one thing that was really fun from my Ph.D. was being able to kind of late in the game, as my interest in evolution kind of continued to grow, to really tap into my friends and colleagues there and to be able to start to think more in evolutionary ways about what some of this research meant. And so by the time I was finishing up my PhD then, I really wanted to move into space where you could do both evolutionary analysis and experiments and kind of use the evolution to test hypotheses and sort of back and forth. And there were a few places doing that, but I thought that Harmit's lab was one of the best, even though he had just opened it only a couple of years before. It was already kind of clear that the trajectory that he was on was pretty interesting. And so I was totally grateful when he offered me a chance to be a postdoc in his lab.
Of course, the work we talked about on, what was it, 198? TWiV 198 was largely work you had done in Harmit's lab, right?
Yeah.
The idea of the expanding genes in the poxvirus genome in response to selection.
Yeah.
I wonder if we could start just chatting about that general idea by having you tell us what a red queen conflict is. 'Cause
Oh,
I don't think we really defined that on TWiV before.
Yeah, so the red queen was actually introduced back in the early 70s by this guy named Lee Van Valen, who is a professor at the University of Chicago. He just passed away a year or two ago.
Did you know him while you were there?
I didn't. We overlapped, but again, I was doing this kind of model systems, genetics, cell biology, and I really wish I would have gotten to the evolution earlier, because it sounds like this guy was just a character from all of the stories I've heard. And in some ways, he was one of the forebearers of open access journals. So he started this journal of evolutionary theory, I think, or something like that, where he just started taking and publishing stuff, his stuff, other people's stuff. And I was just talking to a few people earlier today that reminded me that the journal was actually stapled together. So once they got 4 or 5 papers, they would put them together between a couple of green sheets of paper and staple it and send it out. But so he published this idea of the red queen, and I think it was in 1973 in this journal that he started. The paper is called A New Evolutionary Law. And he was actually thinking more about predator-prey interactions. But this idea, borrowing from Lewis Carroll's character, the Red Queen, from Through the Looking Glass, that it takes all the running you can do to stay in the same place. And so that can apply to predator-prey interactions where, if you're a prey species and a predator can outrun you and hunt you, then you start to select for members of that population that can run a little faster. And then once that happens, it sort of puts the pressure back on the predator to now run a bit faster as well if there are individuals in that population that can do that. And so what's happened then in maybe the last decade is this red queen kind of thinking has really been applied at the molecular level, thinking about host pathogen interactions and specifically host viral interactions especially.
So that's a good description for the poxvirus gene accordion story, right? And it's my understanding that you wanted to, a lot of people do these studies in RNA viruses with a high mutation rate, but you wanted to try it in a DNA virus where the mutation rate is much lower, right?
Yeah, that's right. And so if you think about it, we set up these sort of arms race scenarios as almost a one-to-one thing where you've got let's say, a virus protein that antagonizes a host protein, and then you can go off to the arms races, so to speak. But that's not really how it works, right? So for a pathogen or a virus to be successful, it has to defeat every one of our immune defenses, whereas as hosts, if we just have one defense that can block the pathogen, that's enough. And so it's sort of an asymmetric game in that sense. And so we think that perhaps because of that, that might explain part of the reason why things like small RNA viruses have such high mutation rates. In order to cover against all of those defenses, basically, you have to come up with sort of extreme means to do that. But if you look at a lot of the DNA viruses, like the ones that Tom and I study, the mutation rates are, on average, a lot more modest, orders of magnitude lower in some cases. And so, yeah, that's kind of what motivated our study, was to try to go back and ask, well, what are these guys doing if they don't have these high mutation rates necessarily at their disposal? Are there other mechanisms involved in allowing them to make these moves on the red queen sort of playing field?
So that brings up kind of a tangential question, but
Sure.
you don't mind, right?
No.
Well, if you do, it's just too bad. So RNA viruses have shown that having a high mutation rate can be very useful.
Sure.
They thrive and they spread and they're all over. And DNA-based organisms have this error-correcting mechanism. So why do they have that? Wouldn't it be good to be more mutagenic? And can you test that in a pox virus, which encodes its own polymerase? And I presume an error-correction machinery.
You're right.
Can you take it away?
I think it's a very interesting question. And it may reflect on the differences sort of in strategies that both of these classes of viruses are taking, right? So these DNA viruses oftentimes bring a lot of their own machinery into the game, so to speak, right? So you've got these correcting polymerases, all of these other genes. So in the case of the pox viruses, for example, replication is taking place in the cytoplasm, and so you're not using a lot of the host nuclear factors. You're bringing your own factors. In many of these cases, they are doing jobs that are under tight constraints, where you can't tolerate a lot of mutations in order to carry out those functions. And so in this case, actually having a lower mutation rate is probably more beneficial, potentially, for those sort of core factors, whereas how do you then balance that with needing higher mutation rates with some of the more anti-host defense factors? So the really cool thing about poxviruses is you can almost see that reflected in the organization of the genome, where the center of the genome are all of these conserved factors that are doing those kind of fundamental housekeeping jobs, whereas all of the anti-host stuff, or the stuff going against host defenses, is sitting out on the edges. And in fact, we think that's where most of the recombination-based dynamics, these gene accordions, are happening. And so even how the virus has organized itself may give us a clue into what's going on there.
Is there a reason for that organization?
Well, so it's just a little bit of speculation, but so out on the very edges of the genome are these inverted terminal repeats, these 100 base pair repeats, which are likely to promote recombination. And so the fact that you're just located out there, I mean, it's a little bit, you don't want to go too far, but it could be a little bit analogous to, the centromere versus the telomere, and how you see the difference in recombination out at the edges of the chromosome versus,
Okay.
Yeah.
Didn't we say and add the similar things?
We've talked about this at least, I think, twice before on TWiV.
Which
so
episode?
adenoviruses, I don't know which episode, but I think actually we talked about it on this one, on 198, but another time earlier. Adenoviruses, herpes viruses, it seems like the more flexible parts with respect to genetic evolution are at the ends, and the more conserved parts tend to be in the middle.
Mm-hmm. That's certainly true with the herpes viruses, where the different herpes viruses all have their conserved genes towards the center, and then the specialized genes are out on the ends.
But can you alter a pox virus error correction machinery to make it less, or just get rid of it and see what happens?
Yeah, so I think Paula Trackman has done a little bit of that, and we're following up now in an evolutionary framework. So actually, in addition to those accordion-based mutations, where we saw these rapid gene amplifications of this one specific gene that's out near the edge of the genome, we also saw point mutations that we're following up on now. And one point mutation that we saw is in DNA polymerase. and it wasn't in all of the populations, but it's near fixation in one of the 3 from our first experiment, and then it showed up independently in a second experiment that we did totally differently, different place, different time, different cell line even. We could get the exact same mutation showing again. And so I think this might give us a clue or a hint that, yeah, there might be something going on with either error rate, although I should also point out that the DNA polymerase of poxviruses also has recombinase activity, and so it could be feeding into that, but we don't know, but we're kind of on the case right now.
So as you know, RNA viruses are a quasi-species, right? They're mixtures of non-identical replicons. Is this also the case for DNA viruses?
I don't think it's quite as dramatic. So, you know, with the mutation rates being generally lower, there is certainly variability in these populations, and we see that. And in fact, you also see, you see sort of a dearth of point mutations, but you see quite a few duplications that are seeded at low frequency, even in just if you take a lab strain and look. So yeah.
Okay. So in the pox experiments, which we talked about on TWiV 198, you showed expansion of a gene that counters a host defense protein. That's where you got the accordion name. Was that your idea, the accordion, or Harmit?
Well, it was sort of both. We kicked it around back and forth, and it was a little, I don't know, it was controversial for a while. In fact, in a review, one of our reviewers cautioned us that maybe that was a little bit too provocative of language to include in the title of so that's actually in the title of the paper that came out last summer. And, you know, we kind of punted it and said, well, I think that's an editorial decision. I think we secretly both wanted that in the title.
Yeah.
But, yeah.
I don't think accordion is very provocative.
It's kind of a surprising, seems like a tame word.
Really?
Yeah.
So are you continuing this sort of study in your lab? Are you looking at other genes, other viruses maybe?
Yeah, we are. So right now we're mostly focused on vaccinia and the pox viruses, and there's a lot to do. So even, you know, that arrangement that we were talking about both with herpes viruses and pox viruses, does that really matter? So we're moving around genes that we think would undergo these expansions to see, kind of look at the landscape there. We're also trying to use this as a way to discover sort of new interactions, with the idea that if you just put pressure, in this case we knew, in that first experiment, we knew the host defense gene that the pressure was on. We've now set up different courses where we don't know what the host defense gene is, or what the viral response gene might be. We just have viruses that are really bad at replicating. And then we're looking for these same kind of dynamics to try to discover potentially new interactions. So one really cool thing about the pox viruses is that a lot of their genome, a lot of their genes are actually have their origins as host factors and have been acquired by horizontal transfer. And so we're setting up some experimental evolution protocols in order to see if we can try to get at a little bit of how that process works. How do these viruses pick up host factors? And then once they've picked up a host factor, which presumably is, you know, very little use to the virus, how do those become fixed and potentially beneficial? So we've got a few different lines of investigation going right now, following up.
So getting back to this idea that the genome location is important, if you move this antagonist protein, K3L,
Yeah, that's it.
if you move it from the end to the middle, will it still expand?
That's what we're looking at right now.
You're looking at it?
Yeah, stay
Okay.
tuned.
And then what you were just saying about picking up host genes originally, so do you think K3L, because it's an analog of EIF2 alpha?
That's right.
That it somehow picked that up and then...
Yeah, so the evidence is
Changed
actually pretty clear. So if you just do a blast comparison of EIF2 alpha, the substrate of this antiviral kinase, and K3L, you'll see about 30% identity between the two. And then the crystal structures give even a better view into this homology, where a whole set of beta strands are arranged basically in the identical pattern. And so it's pretty clear in this case that it had K3L was born, so to speak, as a piece of EIF2 alpha. Yeah.
Do other viruses expand the numbers of their genes like this?
Yes, that's a great question. We've been looking like crazy after, you know, that's kind of a good next thing. And I ran into Brian Cullen a week or two ago, and he was reminding me about Epstein-Barr virus and how it has these variable numbers of microRNAs. So I think the canonical number is something like 25. And they look like they could have had a common origin. In fact, it's possible that there could have been sort of accordion-like dynamics going there. So yeah, we're on the lookout, and certainly at least have some clues that and some hints that those things are going to be out there.
How about cell genes?
Yes, so I think that's the case as well. So if you look at some of the host restriction factors, for example, like the TRIM family, TRIM5 alpha is probably the most famous of this anti-SIV, HIV, retroviral restriction factor. There's hundreds of these things in our genomes. And in fact, if we sequenced our genomes, compared the four, I would wager that there are going to be a different copy number of TRIMs among us.
What about RNA viruses? Do you think they would do this?
Yeah, so that's a good question, because here you can imagine that sort of genomic real estate is more expensive, right? You're really highly streamlined.
Right.
And so, but if this happened really rapidly, it might, and we haven't seen a lot of evidence of it, but if it happened really rapidly and was really transient, it is possible. And so I'd say the jury is still out. If I had to place a bet, I might say actually no, but I think it's worth looking a little more closely in some of these cases.
You know, you can make mutations in some RNA virus polymerases that make them more faithful, less error prone. So maybe that would be a good starting point
Oh yeah, it's an interesting
to
idea.
look for expansion when you can't make as many mutations.
Yeah,
Right.
yeah.
But you don't work on RNA viruses, right? So that's
I'm
someone
afraid
else.
of them.
Afraid of them?
I remember you interviewed Terry Dermody not too long ago, right? And he
Yes.
said he picked RNA viruses because he was kind of intimidated by the large DNA viruses. For me, it was the reverse. I'm kind of intimidated by these streamlined RNA viruses that multitask all the time and do all of these things. And the DNA viruses, as a kind of reductionist style biologist, seem a little more tractable to me from a genetic standpoint.
Well, they're not intimidating at all, but they are interesting, I
Yeah.
must say.
Yeah.
What did you talk about at the meeting this morning? You gave a talk, right?
I did. So I talked about some red queen dynamics and tried to do both sides of the arms race. So we talked a little bit about the genomic accordion in the pox viruses, but then also the
So
other.
before you do,
Yeah,
just
please.
for those of the thousands
sure.
here who couldn't
Yeah.
be there this morning, if you could describe, so we've got the expansion, and that gives you now targets of, more targets where the virus could expand or explore mutational space.
Yep.
But then talk about the contraction a little bit.
Yeah, no,
So
thanks,
that's
Kathy.
the part of the
You're right,
accordion.
I should define this a little more. So the finding that we had was when we put selective pressure on the virus, that we had this rapid gene expansion at the K3L locus. We could get up to 20 copies per genome. And there's sort of this 2-for-1 advantage for the virus, in the sense that just having more copies, there's more expression of that protein, which actually gave the virus the ability to replicate better. But what it also did was increase the chance that there would be a beneficial mutation that could arise. This is again all random mutation and selection going on. But by having more copies, you're sampling more mutations. And what we observed was once the virus hit on a non-synonymous change in K3L, the gene that had been amplified in copy number, once this non-synonymous change arose, then under slightly different conditions, the virus started to contract. And so that's the whole point of the idea of the accordion model, is that you expand and contract while gaining that mutation in the middle. And so that kind of shows you how these viruses may have a leg up or an advantage in these kind of arms race scenarios. And then the question becomes, okay, well, what's the host doing? And so that was the second part of my talk this morning, was to ask what's going on with the evolution of this innate immune factor called protein kinase R, or PKR, which is what was antagonizing K3L, or I should say being antagonized by K3L from the virus. And so what we found for PKR was that, so we couldn't do the same sort of experimental evolution that we did with the virus, because you have a problem with generation times in primates and so forth. And so we did a retrospective or phylogenetic analysis among the whole primate lineage. And we found these really interesting signatures of rapid evolution. And so we measure this by comparing the rates of non-synonymous changes to the rates of silent changes. And as that rate of non-synonymous changes starts to get high or overabundant compared to the silent changes, that's a signature of what we call positive selection, or this idea that there's sort of a signature of these arms races. And so the cool thing about PKR is that it looks like there are changes that have been happening on two surfaces of the kinase. One that involves the direct interaction with K3L, the viral factor, but another that's a little distance away. And just single amino acid changes can mean all of the difference between resistance or sensitivity on these two surfaces. And so we think that this gives the host an advantage, where if you're resistant on one of these surfaces, as a population, you can now start to accrue diversity at the other surface. And so if just again by mutation and selection, the virus is able to break through that first layer of resistance on one surface. Now at a population level, you've got more diversity that you've built up in this sort of almost game of hot potato between the two surfaces. And so we think that that's sort of a counter move by the host in order to gain an adaptive advantage in that case. So just kind of putting those two stories together on the pathogen side and then the host side of how we make sort of two moves along these arms race.
So one surface of PKR is interacting with K3L, the vaccinia protein. And what is the other surface that's farther away doing?
Yeah, good question. So it's the E helix, which just sits two helices over in the crystal structure. And it's not clear the direct sort of biochemical link between these two surfaces. But we do have nice evidence that if you just change one amino acid in that helix, that the resistance that you had before to K3L is lost altogether. And so, yeah, I don't have the exact answer for that, and we're kind of looking into that as well.
Yeah, okay.
Yeah.
You also said this morning that there's no comparable change in the EIF2 alpha.
Yeah.
So I don't know if you want to expand on that just a smidgen.
Sure, I'd be happy to. So yeah, that's another, and that's actually what kind of got us excited about the PKR project in the first place. So its substrate, EIF2 alpha, which it phosphorylates as part of its antiviral activity, is under what we call purifying selection. So it hasn't, if you take EIF2 alpha, the variants out of any primate or old world or new world monkey, it's identical. So there's all this constraint on EIF2 alpha. And that makes sense. I mean, it's a translation initiation factor, part of this multi-protein complex. It's highly conserved and is, a key regulator of translation, protein translation. And so from an evolutionary perspective, that's what really captured us, because you've got this kind of sitting duck, EIF2 alpha, that can't change at all. And then this mimic that once was EIF2 alpha that has changed to become this competitive inhibitor. And so in a sense, it just has to target this substrate that's not changing to be effective. And so that puts all of the pressure on PKR, because if it's going to remain a viable antiviral defense, it needs to recognize EIF2 alpha. And so that's really what got us to chase after that one and do the evolution and experimental analysis to find out what was going on there.
So how long have you been in your own lab now?
Yeah, so we're coming up on our 2-year anniversary in July of the Elde lab. We'll be 2 years old.
So you have any advice for people who are thinking of starting their own lab?
I do. So I don't know if it's good advice or not, but it is advice. So the thing that got me really going on it as a postdoc was this feeling as I was going through my postdoc that there are all these ideas out there that we were kind of kicking around and talking about in these really fun projects. And I just kept thinking, I wish I had another set of hands to do this. And then, it kind of dawned on me, oh yeah, people have invented a way of actually doing this. It's called opening a laboratory, right? You get more people, they work with you, you have all these questions. And so, but it was kind of coming at that almost organically and feeling like, wow, I really need a solution to this to chase after all of these ideas and questions. And once I kind of realized that, that was sort of the moment where I realized, I think I might be ready to try to open a lab. And so, so far so good, I guess.
Was it hard for you to find the right position?
It was a challenging process for sure. Yeah. So I ended up, applying to a number of places, had a number of interviews, and in the end, a number of offers, which was a great problem to have. But there are a lot of institutions run in very different ways, and so picking one to go to ended up being pretty challenging, but has turned out great.
So you said before that TWiV saved your life.
I did, yeah. So so how did that happen? Yeah. So when this last spring or last winter, I went on kind of a Mountain West tour. So Salt Lake's about 8 hours from here by car to Denver. But I gave a seminar at the University of Wyoming and a seminar up at Montana State. And so I would just drive to these. And it's really fun because you're in the Mountain West, and there's just beautiful country. You're going through Yellowstone Park. And so driving up to Bozeman, where Montana State is, I wasn't going to fall asleep, but I kind of spaced out a little bit as I was driving up there. And I had my iPhone set, it was no sound right now, the radio off, but I had it kind of in the middle of a TWiV podcast. And somehow it just, I don't know if I jostled it, but all of a sudden there's Rich Condit's voice at like level 60 bellowing out in his baritone about something or other, viruses, and it was like a shot of adrenaline going through my whole system, and I was safe from falling asleep for the rest of the ride. So it's possible that TWiV saved
Sounds
my life.
like it did.
Yeah.
There you go, another advantage to listening to this podcast. You see that, Tom? It's a good thing.
I see it. Yes, yes.
So I have one question. Because you had such a genetic background, it seems to me that you could now consider yourself a card-carrying virologist. Are you comfortable with that?
I am, Kathy. Yeah, I was wondering if that question came up, and I'm ready to own the title of virologist. And so in my talk this morning, as the first slide came up, I noted that I was a virologist in a parasite and bacteria sandwich. And so because the talks before were on eukaryotic parasites and the talks after were on bacteria. So I've announced to the ASM world and the TWiV world now that I do count myself a virologist.
A virologist with a TWiV bump.
We'll see
There
what
you
happens.
go.
I'll keep you posted.
Tom, I know in my early years, you used to publish adenovirus papers, and then suddenly you switched to cytomegalovirus, CMV.
That's right. That's right.
Why did you do that?
Well, that's an interesting question. So at the time that I switched, I had worked with adenovirus for 20 years. And I really enjoyed adenovirus, and I felt like, you could take any gene or any exon within any gene, and I could tell you quite a bit about it. And I started to think, maybe I should start to learn about something different. And, some people, when they want to learn about something different, they go and become maybe a neurobiologist. And I thought about it for a while, and I decided, it would be a lot of fun to learn about a different virus. And so I spent some time thinking about different viruses. Clearly, for me, RNA viruses were off the plate. And so I started to think about the herpes viruses. And at the time that I was interested in picking up a second and a different virus, I became very intrigued by cytomegalovirus because amongst the herpes viruses, it was much less well studied at the molecular level, and there were good reasons for that. But it was much less well studied, and there were lots of opportunities, I thought, to make contributions studying this virus and maybe answering some pretty important questions. One of the things that intrigued me about the virus was that there was relatively little genetics available. People had done mutagenesis analyses using a mutagen, but with adenovirus, we were used to making our own specific mutations at that point, and I thought, hey, I could go to this field and start to make the sorts of mutations I make in adenovirus, and that's going to be great. The problem was that it wasn't quite as easy as I thought it would be. I think it took about 5 years until we could really make mutants in cytomegalovirus. And really the breakthrough was when various people started to clone the virus, the viral genome as a back, and study it and mutate it then in the back. And then you could take the back DNA and transduce it back into human cells and recover the virus and study it that way. So that really works well, and of course many different labs now can make mutant cytomegaloviruses and study the consequences of the mutations.
For those who don't know, BAC stands for?
Bacterial artificial chromosome.
So it's a vector you can put very large inserts into, right?
Right, yeah.
Now, why is CMV worthy of your attention?
Well, CMV actually causes a very serious medical problem. And that is that if a woman is infected during pregnancy, the virus can cross the placenta and infect the unborn child, okay? And the consequences of that can be disastrous. Now, fortunately, the consequences are generally something like hearing loss, but it can range all the way from hearing loss to very profound intellectual deficits that result from this infection. And this is not a rare event. It turns out that cytomegalovirus is the leading cause of by infectious disease of birth defects. So it's a big problem.
But many people are infected with no consequence, is that right?
That's true. So if you look at the literature, people estimate that among adults, the range is somewhere from 60% to 99%. And the range is set by two things. The lower numbers are immunological assays, and the higher numbers tend to be nucleic acid assays, but it also matters where you live, okay? And so if you live in a city like New York, your number is going to be 99%. If you live up in Aspen, you've got a much greater chance that it's only 60% likely that you're going to be infected with the virus. But fortunately, when you're infected by the virus as an adolescent or as an adult, you generally don't even know you've been infected. And so it's not really a problem.
How do you acquire infection?
That's interesting. So you have to think about what happens as the virus infects someone for the first time. And the virus goes through three stages. The first is an acute stage where the virus actively replicates in many different cell types and in many different organs. And then, of course, in a couple of weeks, our adaptive immune system takes over and controls the infection. And then you enter into a persistent phase that can last for months to a couple of years. And during this time, the virus continues to grow in three places that we know of, probably other places that we don't know of, but we know of three. And one is epithelial cells in our salivary gland. One is epithelial cells in the mammary gland. And the other is epithelial cells in the kidneys. And so you end up with the virus in saliva, breast milk, and urine, and that's how the virus makes its way in life.
Okay. So one of the things that you have done with CMV is to look at its effect on the metabolism of host cells. Now, we all know that viruses require the energy that cells make. They need precursors so they can build everything. And so we've known this for years, but you've just gotten interested in it in the past 10 years, maybe. Why is that? Have techniques changed?
Techniques did change, and I met just the right person to work with. So I went to lunch one day with a new faculty member who'd come to the chemistry department in Princeton named Josh Rabinowitz. And Josh and I had a very pleasant lunch together, and he was telling me, hey, I've set up the ability to do metabolomics. And I've been studying the metabolites in E. coli, and I've been studying the metabolites in yeast, and I now know how to measure something like 60 to 80 of them, and I can get a snapshot of what's happening in one of these cells. And that tells me a lot about what the physiology of that cell is in that instant. And so I proceeded to tell him, what I do. And suddenly it occurred to both of us that, hey, he had the technological know-how. I had a viral system, and why not, collaborate and ask what happens to the metabolome of a human cell when it's infected by cytomegalovirus. And I have to tell you that my prediction was that there would be one experiment done and we would move on, because I made the assumption that when cytomegalovirus infected a fibroblast, what would happen is that the virus would consume all of the available metabolic stores in that cell and use those metabolites to make more virus particles. And then when the metabolites were all consumed, the replication cycle would be over and the virus would be ready to go to the next cell. And so what you would do is you would go from a relatively high level of metabolites to very low levels of metabolites. And the cool thing is that we got exactly the opposite result. And this was a set of experiments that was done by a postdoctoral fellow in my lab named Josh Munger, who was working with a graduate student in Josh Rabinowitz's lab. And the two of them together then discovered that when you infect fibroblasts, the level of metabolites, both in glycolysis and in the TCA cycle, almost uniformly increased in amount, okay? And they increased in amount with time after infection. You could first begin to detect an increase at about 24 hours after infection. There were fairly robust increases at 48 and very substantial increases at 72. And for people who are used to RNA viruses, I should explain that the CMV life cycle is very slow. So when we infect primary human fibroblasts in the laboratory, at a multiplicity that will infect all of the cells, we begin to detect the first set of viral genes, the immediate early genes, at about 4 hours. We detect early genes maybe starting at 12 hours. And you start to have DNA replication at 24 hours. And at 48 hours, you're starting to make virus progeny, and at 72, you're peaking.
This is a virus you want to work on?
Hey, lots of time to go home and have dinner, right?
There you go.
So what we found then was that it was just the opposite of what we predicted. The levels of metabolites went up instead of going down.
Can I ask you, before we go on, just name some metabolites so people know roughly what you're talking about.
Well, let me tell you
Okay.
the next step to the story. So one of the key metabolites that we kind of started to focus on was citrate. And citrate levels went way up, okay? And so this is a metabolite in the TCA cycle.
Which none of us remember from biochemistry.
You know, this is kind of funny because the last time I studied any of these compounds was when I was preparing for my prelim exam.
Sure.
And I promised myself I would never, ever think about these things again. So it's funny how everything comes around again.
In a
But
cycle?
when they come around again as part of your science, it's fun, right? But anyway, citrate can be used in the TCA cycle, of course, to generate, as part of the process of generating ATP, but it can also be exported into the cytoplasm, where it is used to make acetyl-CoA, which is then converted to malonyl-CoA, which is then used to make fatty acids. And so one of the things that we thought was really intriguing is that the level of these intermediates that allow you to make fatty acids were all elevated, as was everything else downstream of glucose in glycolysis and in the TCA cycle. By the way, I should point out, one of the interesting things that we found, and I should say Jim Allwine found the same thing at the University of Pennsylvania, was that when the virus infects a primary human cell, it actually converts the metabolism of that cell to look much like the metabolism of a cancer cell. And so the virus is using carbons from glucose, and it's taking those carbons to citrate, and then the citrate is being bled off to make fatty acids. And the TCA cycle is actually depending on glutamine to supply it with carbons, okay? So I think I'm talking way too much and not listening to your question.
It's okay. So you mentioned a couple of metabolites, citrate and glutamine and so forth. And these are measured by what technique that made it possible?
Oh, so yes. So Josh Rabinowitz measures these by a liquid chromatography followed by mass spectrometry.
Okay. And that was the key to being able to do
That was the
all
key to
these
being able
experiments.
to do this.
I think the moral is that you have to do experiments that you think are interesting. In your case, you didn't think it would go far, but it did. Serendipity of science, right?
I think the other moral is that if you can find the right person who has a different skill set than you
Sure.
do, you can make progress that you would never make on your own.
Right.
I remember a story you told a long time ago when you worked on the virus-associated RNAs of adenovirus, and you said that at study section you were sitting next to Chuck Samuel, and that led to a whole series of experiments. And so this is definitely a recurring theme in your science that I've heard about.
Yes, Chuck Samuels, of course, was and is Mr. Interferon and all of the pathways that it can influence. And that was very important back then when we were studying VARNA and trying to find what it might be doing, and we eventually discovered that it had a lot to do with PKR.
Yeah.
All right, back to the metabolomics. This is really interesting. So the CMV increases the overall metabolomic level, which is contrary to what you thought would happen. And a major aspect is lipid biosynthesis. So one of the questions is why? Do you know why it's increasing these things?
I partly know why.
Okay.
And this now is work that was done by a fellow named Emery Kuyuntsu, who's a postdoc in my group, and John Purdy, who's a postdoc with Josh Rabinowitz. In fact, the paper has just come out. And what Emery did was to do an siRNA screen where he screened a bit more than 400 enzymes and regulatory proteins that are focused on metabolism. And when he did this screen, of course, whenever you do these screens, you get lots of hits, but he got two sets of hits that were really interesting to us. The first were hits in a family of proteins called acyl-CoA synthases. And so these are proteins that can take a fatty acid and activate the fatty acid. And the second family were elongases. And elongases can take fatty acids that are at least 14 carbons long, and then in 2 carbon units, they can increase the length of the chains.
So these were required for viral replication.
Yeah, and so when he did the siRNA screen and knocked them down, that reduced the yield of the virus. So this then focused us on the production of very long-chain fatty acids, which is totally consistent with the need to make malonyl-CoA, which I had mentioned before, because malonyl-CoA is what is used by the elongases to elongate a fatty acid 2 carbons at a time. And as you can imagine, since this has to do with lipid biosynthesis, there's been quite a few candidate drugs developed and studied by the pharmaceutical industry, so it wasn't too hard to purchase and also to beg for compounds that block the activity of these enzymes. And the result was quite interesting, and that is that you made less progeny virus particles, and the progeny virus particles that you made were less infectious. And this wasn't surprising to us because, of course, cytomegalovirus, like all the herpes viruses, has an envelope, so it has lipids in the virus particle. And we're now messing with the fatty acids, which would be potentially a component then of the lipids in the virus particles. And then this is where a beautiful experiment from John Purdy played into the show. And John developed the ability to use mass spectrometry to analyze the lengths of the chains and the saturation states of the fatty acids in a cell or in a virus particle. And what he found was that in a cell, if you looked late after infection, there were more of these very long-chain fatty acids present than in a normal uninfected cell. So that fit beautifully with what we were seeing in terms of a requirement for these enzymes that could make that happen. But the real payoff came when John looked at cytomegalovirus particles, and in those particles, they were very heavily enriched for these very long-chain fatty acids, 26 carbons all the way up to 34 carbons. We can't measure things larger than that. And so now we know that the virus needs to make the malonyl-CoA. It needs the enzymes that will allow malonyl-CoA to be built into very long-chain fatty acids. And we know that if you don't make those very long-chain fatty acids because you treat with a drug that inhibits those enzymes, you make virus particles that have reduced infectivity. Okay. So we're kind of pretty much focused now on what is it that is important about very long-chain fatty acids that the virus needs that in its envelope. And the answer is, the answer to why the virus needs that is that we don't yet know. There's a literature that says that if very long-chain fatty acids are concentrated on the inner leaflet of a membrane, that it encourages curvature of that leaflet. And so you could imagine that this is important for curvature of the virus's envelope. But unfortunately, for that idea, if you go and look at herpes simplex virus and look at its envelope, it ain't got these very long-chain fatty acids in it. So all viruses don't need to do this. I mean, maybe cytomegalovirus does.
So these acyl-CoA transferases are not required for herpes simplex replication then?
That's correct. That's correct. We've actually assayed that. A graduate student named Sarah Grady has done that screen, and they didn't come up in that screen.
But presumably the fatty acid requirement is one part of this whole metabolomic increase, and there are many other metabolites that are needed. But do you understand why as much as you do for the fatty acids?
Well, I think some of the other changes are more obvious. For example, the viruses upregulate the synthesis of all of the building blocks for nucleic acids. But that's pretty easy to understand. It seems straightforward. There's many changes that probably make sense. I mean, the virus needs lots of ATP to be available in those cells because those cells are being stressed to produce lots and lots of virus progeny. So I think a lot of the changes make pretty good sense. And the ones that really intrigued us was over in the lipid biosynthesis.
Okay. Are these therapeutically useful, some of these targets?
I would predict that they could be. And as a disclaimer, I consult for a company that's working on exactly this. But if you think about it, if you can interfere with the ability of the virus to either make an envelope or to make an optimal envelope that is optimally then infectious, that could be big. And I think that there are potentially two advantages. One is that I'm going to guess that at least amongst the herpes, there's going to be some other herpes viruses that need this, and maybe some other viruses that have envelopes as well. So they might be broad spectrum. Okay. But the other thing that I think is really intriguing, if you go after the virus this way, instead of going after it by creating more direct-acting antivirals, is that it should be harder for mutant viruses to accumulate that can bypass the block. And so I think it's an interesting area to explore and see what happens.
So most of this work is done in cell culture, infected with viruses, right? Is this safe to assume that because these are fundamental metabolic pathways that it would be the same in an animal? Or do you think there might be differences?
So that's a very interesting question, because if you say, Tom, would you assume that it would be the same in different cell types? I would say no, I wouldn't assume that at all, because we know that there are some other cell types where the virus can infect and enter, but we don't see these dramatic changes. One cell type being a monocyte. And the interesting thing about the biology of a monocyte relative to cytomegalovirus is that when cytomegalovirus enters those cells, it doesn't replicate lytically, but it enters latency, so it becomes quiescent. And when it infects those cells, for a brief period of time, it expresses lots of its RNAs, but then everything shuts down. And during that brief period of time when it's expressing lots of its RNAs, metabolism doesn't change a whit in those cells.
Okay. Let's get a question from an audience member here.
Tom, that's a very interesting story about the very long-chain fatty acids in the envelope of CMV. What surprises me is envelopes of viruses have been studied for a long time. I would have expected that people would have said, well, this is really unusual that we have these very long-chain fatty acyl components of the envelope. Did people just not look? And if you're going to predict that there may be this broad spectrum, could you guess which viruses by asking which viruses have these very long-chain fatty acids in the lipids of their membranes?
So Stan, that's a great question, and I think you're right. People just haven't been looking. Although there is a literature in HIV where people have reported that they can identify very long-chain fatty acids present in the HIV envelope. You know, one of the things that I think the abnormal, as I might call it, lipid composition of the viral envelope is telling us is that cytomegalovirus is acquiring that envelope at a specialized membrane within the cell, okay? And so maybe the way to think about it is to ask, well, what is the set of viruses that might be maturing at that particular membrane within the cell? And part of the problem with pursuing that question is that we still don't know what membrane within the cell the virus matures at. There's a lot of, gee, just maybe this is where it's maturing, but that's a really hard point to nail. But you're right, it would be very interesting to know the full sets of viruses that might have these very long-chain fatty acids.
So back to the other question. You could take a human whose salivary glands are secreting CMV and take some cells and do metabolomics on those, I presume. Is that worth doing?
I think that that is very worth doing. And I think that it is an experiment that is doable, but it's quite challenging. Because if you do anything to perturb a dish of cultured cells, for example, if you decide, well, I'm going to measure the metabolism of these cells, so I'm going to remove the medium, and then I'm going to wash them once with buffered saline, you've substantially changed the pools of metabolites in those cells, and no matter how fast you do it.
Yeah.
And so when we do these experiments, what Josh's lab figured out was that you just remove the medium and then you go with it, okay? And you can't spend time washing it or you're going to change what's happening. So the challenge then is to harvest the salivary glands and analyze them without perturbing metabolism.
Right.
I think it's doable, but it's not trivial.
Yeah. So how do the viruses affect this change of the metabolome?
Yeah, that's a very good question, and we have some insights, and there's lots that we don't know about it. So Josh Munger has focused quite a bit on that, and one of the targets that he found, and I should say we found it as well, is AMP kinase, okay? And so, you know, AMP kinase can be activated by stress.
This is adenosine monophosphate. Yes?
Yes.
Okay.
And this kinase can be activated by stress. And when it's activated, the kinase can activate fructose bis, let's see, PFK, phosphofructose kinase, and it can also activate acetyl-CoA carboxylase. It inactivates acetyl-CoA carboxylase. And so both of those enzymes are key enzymes in the changes that we see because phosphofructokinase is a rate-limiting enzyme in glycolysis, and AMPK is upregulating that, so that all makes beautiful sense. Acetyl-CoA carboxylase, we know it's upregulated, we know it's required, we know it's phosphorylated by AMPK, but it's somehow being kept active by the virus, and we don't know how. And I think that's a really interesting question that you know we need to explore further. But certainly AMPK is helping to activate glycolysis by activating that specific enzyme.
Okay.
Another thing that Jim Allwine showed was that a glucose transporter, GLUT4, is very dramatically and very substantially induced by the virus when it infects a cell. And of course, you need to import the glucose to start the whole show here. And so I think that was a very critical, important observation. That's a very early event. It's triggered by a very early event in the virus life cycle. We need to know more about how it's triggered. We don't know exactly yet how it's triggered, but that's something else that helps to initiate this increase in metabolic flux.
So the virus is tapping into existing pathways that the cell would normally use for various reasons. And the virus has evolved to be able to do that, essentially. It's not inventing anything.
That's pretty much what the virus always does,
Yes,
isn't it?
it is.
It tweaks here and manipulates there and gets the cell to do what it needs.
Right. So every virus on the planet is going to be messing with the metabolome in some way.
I would predict that that's very likely true. Now, only a few viruses have been looked at so far,
Right.
but each of the ones that we've looked at are turning out to do things. So I think it's likely.
So you've looked at a few herpes viruses, right?
Yeah, we looked at cytomegalovirus, we looked at herpes simplex virus, and we also looked at influenza A.
Okay. And do you know of other viruses that are being looked at elsewhere?
Well, I'm sure lots of viruses
Yeah.
are being looked at now, but
Okay.
So, and just to make it clear that I'm guessing that these different, these changes in metabolomics are different among CMV, herpes, and influenza, because you even alluded to it being different in cell types.
yes, they are different. So each virus appears to establish its own program of metabolism. So that's actually, to my mind, a very interesting result, because that says you're not just looking at a cellular response to infection. Part of it might be that, but a lot of it is virus-specific manipulation of what's going on. And in simplex, the biggest changes you see are not in anything to do with the production of fatty acids. It has to do with the production of the building blocks for nucleic acids. And that kind of fits with the idea that this virus very quickly institutes a very massive program of DNA replication. And CMV, on the other hand, takes a long, long time to get around to it.
So, and my thinking was that adenovirus being non-enveloped would also probably not make as many changes to the fatty acid part of the metabolome.
That's right. We've taken a very preliminary look at adenovirus, and we plan to look a lot more, but it does institute changes, but nothing in terms of the fatty acids.
Back to adenovirus.
Yeah.
You can't avoid it.
Well, you know, I'm lucky. Jane Flint is right down the hall
Yeah.
from me, so I can always go to Jane and say, hey, could I have this or that or the other thing? She's got it all going.
Did you know this story, Nels?
Yeah, a little bit,
cool.
and actually, it's interesting. So Tom's former postdoc, now professor at University of Rochester, Josh Munger, he and I were classmates together, actually, at the University of Chicago. He was with Bernard Roisman, and I was with this fellow, Aaron Turkowitz. And so it's funny, the kind of connections, right, if you just sit down with a fellow scientist.
It is.
Usually about 1 degree of separation.
Yeah, right. Yeah.
So did you speak about this morning at your talk?
So I talked about something that is kind of an offshoot of it, and that is the fact that sirtuins are viral restriction factors.
Sirtuins, these are the things that make you live forever, is that right?
That's right. These are the things that resveratrol, which is in red wine, activate. Okay, and so resveratrol could be a very good thing, and certainly I'm willing to assume that it is. But it's funny, we started working on this again because of a conversation, this one just in the hallway, and this was a conversation I was having with Ileana Christie, who is the faculty member across the hall, and she's a mass spec expert, but she does proteomics. And we've collaborated quite a bit through the years. She's just terrific. And one of the things that she has been very interested in is HDACs. And she's studied and generated the interactome of the 11 HDACs.
So those are histone deacetylases.
Yes, I apologize.
Yeah, I
So
was gonna
HDACs are histone deacetylases. Thank you. And in that family, there's another group of 7 histone deacetylases, or protein deacetylases, and they're called sirtuins. Okay, so they're officially class 3 HDACs. Okay, and the thing about the sirtuins that we talked about that day was that the 11 HDACs that she studies operate at one level independently of the metabolic state of the cell. When you compare them to sirtuins, because sirtuins are NAD dependent. And so NAD, of course, is a critical metabolite. And as NAD levels rise or decrease, that then impacts on the activity of sirtuins. Okay. So there's 7 sirtuins. They're connected with metabolism through their NAD dependence. And actually, they're protein deacetylases. They're demyelinases, succinylase, and myristoylases. So they can remove a variety of different modifications from proteins. And so that day, Ileana and I decided, hey, why don't we do a collaboration? Tom, you've gotten pretty good folks in your lab in running these siRNA screens, and I know everything about HDACs, and so why don't we run the 7 sirtuins against cytomegalovirus and see what happens? And the surprising thing was that if we knocked down with the siRNA any one of the 7 human sirtuins, the yield of cytomegalovirus went up. And in that experiment, it went up anywhere from about 3-fold to maybe 10-fold. Okay. So that suggested then that the sirtuins are viral restriction factors. And we went on to find the same thing happened with herpes simplex virus. The same thing happened with adenovirus, and the same thing happened with influenza. And amazingly, if you move to lambda in E. coli has one NAD-dependent protein deacetylase. It's called COB-B. And so if you make a pair of isogenic E. colis, one has it and one doesn't have COB-B, lambda generates a larger burst size in the absence of COB-B. So that raises the possibility, as we were talking before, we went live, that it's conceivable that this virus restriction system is really ancient and goes all the way from mammals all the way back to E. coli. Of course, we've not looked at anything in between those two extremes.
So would you say that most eukaryotic viruses would be sensitive to them as well?
What I can say is that I know that the 4 we
Or,
tested
yeah.
are.
You certainly won't look at more RNA viruses, right?
I think that would be a good idea because, you know, lots of RNA viruses are pretty important.
Yeah. So do we know how the sirtuins impact, or how they are restriction factors? I see you've taken on the nomenclature of the retrovirologists now. You're calling them restriction factors. How do they work? How do the sirtuins impact?
So we've looked at a couple of them, but we're just beginning to scratch the surface here. And the first 2 that we looked at was kind of by accident because there was a drug that you could buy. So I told you if you knock down sirtuin 1 by itself, the virus grows better. Or if you knock down sirtuin 2 by itself, the virus grows better. And actually, if you use an agonist that increases the activity of sirtuin 1, the virus is toast. Okay, so it can't grow then. But we also were able to purchase a compound called Cambinol. And Cambinol is a dual sirtuin 1-2 inhibitor. Okay? So you might have expected that the virus would grow really well if you treat cells with cambinol, but cambinol turned out to block the growth of cytomegalovirus and also blocks the growth of influenza. So what's going on here? Well, cambinol has generated a lot of interest because it actually has activity against P53. It turns out that sirtuin-1 and sirtuin-2 each deacetylate one lysine residue and a different lysine residue on P53. And when the two sirtuins are active and they're deacetylating the P53, that reduces its activity, okay? And when P53 is hyperacetylated at those two lysines, it is highly active, okay? So that's one fact. The other fact that we knew was that in cytomegalovirus infected cells, P53 is there. It accumulates to fairly high levels, but it's inactive. Does that sound familiar, Kathy? Yeah, the same thing happens in adenovirus, and influenza does the same thing. And so what we believe then is happening is that cambinol is blocking the activity of SIRT1 and 2. They stop deacetylating P53, and so P53 becomes hyperacetylated and active, and that then interferes with the growth of the virus. Okay.
Because the active P53 is blocking the cell cycle progression?
Well, I don't think cell cycle progression is necessarily important to cytomegalovirus. The fact of the matter is I don't quite know what the block is in cytomegalovirus, but I do know that it's P53 dependent, because if we knock down P53 in a cell and then infect it with cytomegalovirus, the drug no longer has this effect.
Right.
Okay? So I know that P53 is important, but whether it's cell cycle or whether it's apoptosis or whether it's something else, we don't know yet. We do know for flu that in the presence of the drug, the infected cells immediately become profoundly apoptotic. And so I think in that case, it's inducing apoptosis before flu is ready for it to happen.
So we should probably say that P53 is a tumor suppressor protein that is involved in mediating the progression through the cell cycle, depending on what the conditions are in the cell, right?
You said that very well, Vince.
Thank you. Just taught it in my class a few weeks ago. That's a great story. And it sounds
I
like...
think it's interesting, and there's so much to do.
Yes,
we're
clearly.
just beginning to understand.
And you've given other people a lot of ideas as well.
I hope so.
So the moral is you have to have good colleagues, and you need to talk with them. You can't just stay in your office and not speak with anyone. You got that, Nels?
I'm taking notes over here.
That is tremendous. Well, I think we should wrap up this episode of TWiV, which, like all the others, will be found on iTunes at twiv.tv. And if you like what we do, one of the things you can do to help us is to go over to iTunes and leave a rating for the show, which keeps it more visible so that more people can find out about the cool world of viruses. And of course, we love to get your questions and comments, and we usually answer them on other TWiV episodes. You can send them to twiv at twiv.tv. Nels Elde is at the University of Utah. He has a pretty cool website you should go check out.
I agree.
Thanks for joining
Check it
us.
out.
Do you do that yourself, or people at your lab maintain?
I do, actually. So, yeah, it's a little bit of a hobby, I guess.
Yeah, he has lots of cool links. He's divided up his lab into different areas and graphics, so go check it out. We'll put a link to that in the show notes. Anyway,
Great.
Thanks for joining us today.
Thank you.
Appreciate
Very
it.
fun.
Tom Shenk is at Princeton University. Thank you for joining us.
Thank you, Vince. It was great to be here.
Your first podcast, now you're over the hump, and maybe you'll get a TWiV bump.
I hope so. And maybe you'll invite me back someday.
But I don't think you need a bump. You're doing quite fine as it is, right? Yes, we certainly will have you back. Kathy Spindler is at the University of Michigan. Thanks for coming by.
Thanks. It's always fun.
Your first live TWiV.
It is.
It was live.
It was live.
Many thousands of people. I'm Vincent Racaniello, and you can find me at virology.ws. I want to thank the American Society for Microbiology for having us here at the general meeting. Our producer, Chris Kandayan, who is running the TriCaster back there. Is he still there? He's hiding. There you go. There's his arm. And Ray and Andrea on the cameras. Thanks a lot. And this nice audience, thanks for coming here. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.
Once you're in the BSL-4 space, the only way you can get out is to go through a chemical shock.
It's an unusual room. Never seen anything like this.
Anybody who has access to this facility first has to go through an IR scan.
So the HEPA filters filter the air coming out of the facility, and that will remove bacteria, viruses, anything that might constitute any kind of risk, right?
Remember, this building is basically a second building inside the main building.